Welcome my students. Today in the BioClub activity we are examining the plant cell, and in this case we have the onion bulb. First of all, we will use the compound light microscope, and in the previous session we illustrated the structure of the compound light microscope. So now we are preparing the slide to be examined. First we have the plant, which is in this case the onion bulbs, and here we have a leaf. In order to examine it first, we will use the internal or the inner epidermis of the leaf, and firstly we have to handle it and prepare it for the examination. So I'm using the scalpel to open it, cut it open. Afterwards, there will be the inner epidermis. Okay. Here is the inner epidermis. Beforehand, I will place a drop of distilled water on the slide in order to maintain the osmoregulation and the turgor pressure. And as you know, the distilled water, in comparison to the water inside the plant, it has more water potential. So, as you will see now, I take off the inner epidermis layer and I place it on the cover, on the slide, and on the slide I added a drop of distilled water. Why? Because now, as you will see, you will observe, the water of the water potential of the distilled water is higher than the water potential inside the plant leaf. So the water will moving from a region of their high water potential, which is outside in the distilled water, to a region of their lower water potential, which is inside the plant leaf or the epidermal cells, by process of osmosis. Okay, and this play a very important role, making the cell more turgor, increases the turgor pressure, which is really, really helpful while we are examining the plant cell. So now the cell is taking up the water by osmosis. Okay, the water is moving from the distilled water surrounding outside into the inner cells, which is the inside the plant leaf by the process of osmosis. Okay. So after a couple of seconds, the osmosis will be accomplished, and afterward we will remove the excess amount of water using this tissue. Okay, just from a side, I'm going to add in the tissue. The tissue will absorb the excess amount of water. Afterward, I like to handle it for the examination. In order to obviously see the nucleus, I'm going to use a safranin stain in order to stain the nuclei, to be obvious and we can see it properly. So just one drop of the safranin stain, I'm going to add in it on on our specimen, and now the cells will taken up the stain and the nuclei will be clear. Okay, for just a couple of seconds in order to allow all of the cells taking up the stain. Afterwards, okay, I will wash the specimen by distilled water in order to rinse the excess safranin stain. I'm going to add in another drop in order to be, okay, to acquiring the stain. So I can tell now that the nuclei taking up the safranin stain. In order to rinse it, I'm going to do this here in the Petri dish. Okay. Or firstly, anyway, I'm going to using water. So now the excess amount of safranin is being rinsed out, and I'm now sure that the nuclei taking up the the safranine stain. And also the distilled water play a very important role in maintaining the osmoregulation of the plant cell. Okay, that maintaining the turgidity of the cell and making it very easy for us to examine it. Okay, okay. Now I can tell that the excess amount of the safranine stain is being, okay, rinsed out. So same tissue for another one here, another tip. So now the excess amount of safranine and even the water is being rinsed or cleared. So now in order to examine it, to see the cell wall properly, I'm going to use another dye which is known as methylene blue. Methylene blue. Yes, here I'm adding a drop. Okay, there is a bubble. I have to avoid bubbles of the methylene blue. Yes. And I just leave it for just a couple of seconds for the plant cell to take it up. Afterward, I'm going to use a new clean tissue. And this is one of the tricks in order to prepare it properly. Just from a side, okay, place the tissue it will take in it. But in order to avoid the bubbles, I will use another, okay, tech in order to be sure that there are no bubbles. The cover slip, we are using it. And in order to avoid the bubbles, we will make it in an acute angle, which is about 45 degrees, in order not to trap any bubbles. And gently, we are downing or moving down the cover slip, okay? And be sure there are no air bubbles trapped, because the air bubbles make the vision unclear. Yes. Now I can tell that roughly there are minimum amount, or now there are no, okay, bubbles trapped. In order to take out the excess amount of the methylene blue, I will just place the tissue to one side of the specimen. Then all of the excess amount of stain will be taken away. Now I can say that the specimen is ready to be examined. Now, and even to recall what we just learned in the process of examining the specimen under the light microscope, again, here. Now we have the light microscope. I'm going to place in the specimen on the stage and securing it using a stage clip, and afterward I will brought the magnification power to the lowest, okay, and I'll be sure that the specimen is right under the lens straight away, and I'm using a handle in order to move the stage forward and backward and slide right and left. Now I can tell that it is perfectly, okay, fitted, and I will now raising up the stage using the coarse adjustment to the top, okay. With this stick, I'm know now that the stage is no longer can move upward, okay. Now I'm going to looking at the eyepiece using the eye lens. Now I can see very clearly the cells under the microscope. Afterward, I'm going to use the fine adjustment for better viewing the cells. Now the cells are really, okay, perfectly seen under the light microscope. I'm looking on my field for a very clear vision in order to let you see it and view it. Yes, now it's really nice. Yes, here is awesome. Yeah, that's very awesome. So now I can raise the magnification power to the next level. I use the 4 or the magnification power of 4. I can now move the objective lens to the next magnification power, which is here, the 10. And I will not touch the coarse adjustment in this case. I will just move the fine adjustment, okay, a little bit for adjustment of the resolution. Now the cells are obvious. I can control the amount of light for better vision, okay, decreasing it or increasing it according to my situation. Here I'm using the maximum light intensity. I will reduce it a little bit in order to have a clearer, sharper image. Now I decreased the light intensity and focused it on the slide, okay. Now I can tell that the vision now is really, really clear. You can now come and see the plant cells under the compound light microscope. The plant cells are really, really beautiful. They are very clear. You're gonna see even the cell wall, and there are no bubbles trapped. Okay, so one by one, you can come and tell me what you are seeing under the microscope, and I'll take up your comments. Yes. For those that are not in our biology club, I will share you the picture that we are actually examining now under the compound light microscope. Okay, what's your name?
Abdussalam.
Okay. What are you seeing now under the light microscope, Abdussalam?
I'm seeing a bunch of lines, and it looks like cell walls, like little bricks.
Yeah, exactly. Brick-like structure.
Yeah, brick-like structure.
Yeah. It looks like rectangular a little bit, isn't it?
Yes.
Exactly.
And there's some dots. There's some dots there.
Exactly. The dots are the nuclei, and you can see clearly the cell wall. I think it's very clear. Could you see the cell wall? The limited line, or the line that's been limiting the plant cell and separating it from the adjacent cells.
The black lines?
Yes, exactly.
Yeah,
the dark lines.
Oh, yes.
Yeah, thank you.
See that.
Thank you very much, my dear. Yes, Sam? Yes, thank you. Have a look. What's your name first?
Saad Luthman.
Okay, that's great, Saad. What you saw under the light microscope?
I saw like a brick-like structure, and I think it's palisade cells.
Yeah, great. What is the function of the palisade cells, Saad?
They are the key, or the most important role in photosynthesis.
Exactly. This is the most photosynthetic tissue. Yeah, it is the top under the surface of the. Yeah, thank you. Thank you. Carry on. Okay, thank you very much. So, my students, today we examine together the plant cell, and in which we together prepare a specimen from a plant cell. Our example for today was the onion bulb, and I hope you enjoyed the activity of the Bike Club today. See you inshallah next time. Till then, stay safe and stay blessed. Have a good day.